So was told that we should do a broad introduction. So I have copied and pasted the talk that I gave for ISMB into the beginning of the lab meeting. So it'll be great that it's recorded, and you guys who have probably never seen this before will see it, and everybody else can play with their phones or something.
Estimate people's retention, I think. Anyway.
Okay, so the talk is enabling biological knowledge integration and curation through Wikidata. So I start with this hypothetical question that a researcher or scientist may be interested in answering. The question is, can you show me drugs that interact with a protein, drugs that interact with a protein, where that protein's gene is associated with a disease, and limit it to proteins that localize to the cell membrane and diseases that are respiratory diseases. So this is a pretty straightforward question, but it can be really difficult to answer for many reasons. One is unstructured data. So there's a lot of data that exists only in the literature. If it is in databases, it can be scattered across many different databases or resources. So there's drug data from lots of different places, or from drug.info. There's gene, protein information, gene disease associations from like PhenoCarta or GWAS, PhenoScanner. There's many different disease ontologies, UMLS, disease ontology, Monarch. So to answer this, it would require accessing at least 4 different databases, dealing with file formats, APIs, licenses, identifiers. One of the big issues is that many of these biological entities have different identifiers from different resources. So as a really common example, metformin, which is a kind of drug, has at least 11 different identifiers from different databases. And when you want to normalize these things together to look up, for example, drug-protein interactions from other databases, you have to normalize all these identifiers together. So this hampers interoperability, makes scientists' jobs more difficult. So one solution, potential solution for this problem is we can approach with Wikidata. What is Wikidata? It's to data as Wikipedia is to text. It's a collaboratively edited knowledge base operated by the Wikimedia Foundation. It's free, even for commercial use, so it has a CC0 public domain license. Anybody can add, contribute data to it. Covers all domains of knowledge, not just biological data. So it's everything that's in Wikipedia. It has, just like Wikipedia, each item in Wikidata has item history, talk pages, there are projects, user accounts. It's integrated within the semantic web through this query engine, a Sparkle query engine that's used for querying ungraphed databases. It is stable, so the long-term support is not dictated by local funding cycles. It's supported by the Wikimedia Foundation. It's being actively developed by them, and it already has a large number of active users, editors, and contributors. So the way data can be visualized in Wikidata, or the way data can be added onto items in Wikidata are through statements on items. So as an example, this is a protein item in Wikidata. It has, each item has a unique identifier, which is a QID. Each item can have labels, descriptions, and aliases in any number of languages. You have a question? Oh, no, okay.
You know what he thinks.
You're like looking at him. You're right in front of my view, so I was looking at you, I'm sorry. Each item has a list of statements where a statement is composed of a property, a value, and an optional list of qualifiers and references. So for example, retinoic acid receptor alpha has a molecular function, retinoic acid binding, and it has a determination method, IDA, inferred by direct assay, and then it has a reference saying when it was retrieved, where this annotation came from, what the curator was, and a reference URL back to the original data source. And each of these things in here are items in Wikidata as well. So retinoic acid binding is an item, and it has a QID, and it itself has a list of statements. So for example, it's a subclass of retinoid binding, and all of these things that are used in these statements are all items, so everything is one big graph. In addition, Wikidata is also used as sort of a hub for the various language Wikipedia articles. So retinoic acid receptor alpha English Wikipedia articles and the Spanish Wikipedia articles, et cetera, are all linked together through this Wikidata item. As I said, there are item history, so each edit is tracked with a date and user, and so you can link to specific revisions with a unique ID, and there are talk pages as well. So what we've done is modeled the structure of various items in Wikidata that I'll talk about a little more on the next slide. Imported data from many different sources and ontologies, reconciled many identifiers from external databases, and I'll talk a little bit about the next couple things also. So this is a sort of an overview of all of the biological entities in Wikidata and how they're connected to each other. There's actually more than this, but these are the major ones in a much more easily digestible graph. So we have genes, proteins, genes encode proteins. Proteins are part of protein families. They can physically, or I guess this should be the other way around. The chemical compounds can physically interact with proteins. They can both be part of pathways. We also have genes that have genetic associations to diseases, have drugs that can be used for treatment of those diseases. And then these are also linked together with variants as well. And then some of these entities have outside links into the rest of Wikidata as well. So what can you do with this? You can do, so I'm going to build up to that initial question from the first slide, sort of step by step, that I kind of stole from Andrew, because it's a good example. So you can start with simple data retrieval. So for example, if I want to just retrieve genes with a genetic association to asthma, all I would need to do is write a really simple Sparkle query. So this is the Sparkle query language down here. What I've done here is just to make the query a little more legible, instead of using, so as I said, each item and property has a QID and a PID, and instead of memorizing the QIDs and PIDs, I've just given them names here. So P2293 has genetic association, and Q35 whatever is the item for asthma. So you can just select genes and their labels where a gene has a genetic association to asthma. That's basically the whole thing, or that is the whole thing. And you get back a list of genes, and this is an example of the statement that it's actually getting this from. So this is a statement on one of these gene items that has genetic association to asthma. And so you can go back to the original data and look at it if you'd like. You can then take it a step further. You can leverage ontologies within this query. So we can say, restrict it to genes whose gene product is localized to the membrane. So the membrane is a gene ontology term it's a cell component within the gene ontology. And a protein may not be annotated exactly as membrane. It may be to a term more specific than membrane. So we can actually query for all of those at once. So we can say so this gene encodes a protein where each of these things have PIDs and QIDs. That protein has a cellular component. And then this means something that is a part of or subclass of membrane. And the star means any number of steps. So any term that is descendant through part of or subclass of membrane, and then get any proteins that have cellular component annotated as any one of those terms. And we get back an answer. We can even compute on the provenance itself. So we can say something like, only show me annotations that are not electronic annotations. So we have like the determination method in here, and you can query on determination method or anything in the reference or anything like that if you'd like to. And then I took out a couple slides, and now you can answer this entire question in just really 6 lines. So get me genes that have genetic association with any disease that is a subclass of respiratory disease. So this takes advantage of the disease ontology. Gene encodes a protein, that protein's statement, get the value, it's part of or subclass membrane, and then filter out any statements for the determination method is IDA, and then get me drugs that physically interact with that protein. And you get back, for example, rolopram, which is a phosphodiesterase inhibitor that localizes to the membrane, and it has a GWAS association with asthma, which is a type of respiratory disease. All right, so yeah, so as I mentioned earlier, so Wikidata is a source for information, but it's openly editable, so people can add to and update Wikidata as well. And one of the things that are highly active are diseases. So we have a project with the disease ontology where we've imported all 6,000 diseases from the disease ontology into Wikidata. This was like 2 years ago almost. And then let people do whatever they wanted to these diseases, add or update or change statements. And then the idea was to take some of this crowdsourced knowledge from Wikidata and filter it and prioritize it through GitHub tickets for curation by the disease ontology curators, and then statements or knowledge taken from Wikidata that have been added by others, that have been vetted by the disease ontology curation team, can then get updated in disease ontology and pushed back into Wikidata as well. And so you have this sort of feedback loop where we take crowdsourced annotations and then curate them and integrate them into primary resources and cycle it back through. So this is a sort of summary on the activity of disease items. So the red, this is just a pie chart of, there's 8,000 diseases. About a quarter of them had some sort of edit on it over the past 1.5 years, and 3/4 of them were untouched. That represents over 2,000 diseases that had some sort of crowdsourced edit or action on them. Most common was addition of cross-references. So there were 1,400 new GARD, which is a rare disease database, identifiers added to disease items that weren't there before, about 500 OMIM, 400 ORPHANET, 300 MESH, 65 UMLS, cross-references, they're all added. In addition, there were edits, so cross-references were fixed on tens of items. So, just Wikidata's found tons of errors within the cross-references in disease ontology. And they also added or edited many subclass of links also. So they're actually editing the ontology itself.
Are they typically user class edits or the user manual edits? Can you tell?
So for guard, it was mostly done through mix and match, which is, I think it's by hand, but it's through a tool that allows you to like quickly match up things by label. So if the label matches, it will like propose that as an addition and you can just click accept. Just click accept. And that's, I don't know how many of the 1400, but that was done with guard. These other ones, I think were mainly done by hand, although I can't really know for sure. So yeah, and other people have asked also, so like who's actually doing this? So this is a plot of number of edits per user. And you can see some of these are bots, but also sometimes you can use a bot, you can write a bot and use your user account to run the bot. So the account may not be marked as a bot, but you could still automate it. So there's no way to really tell. But you can see there are some really, really super active users that have tons of edits. And then, so there were about 405 unique users that did some sort of edit on a disease item. And so you have like maybe the first, it was like the top 20 users did like 80% of the edits or something like that. But you have hundreds of other users who each made 1 or 2 edits. All right. Okay, so one of the challenges that we have in Wikidata is how do we get more researchers to use and contribute to data in Wikidata? And one of the ways we could do that is to build targeted communities that on top of Wikidata to allow researchers or users to use it for targeted knowledge integration or curation. And so one of these that's being developed in the Sioux Lab is Clanbase, which is just like some of these other sites are actually they're applications that can use Wikidata through the Sparkle endpoint to retrieve or edit data. So this is Clanbase is basically a genome browser with many other annotations that are accessible through it. So Clanbase is targeted to the chlamydia research community. And I forgot what I was going to say. Yeah, so the idea is that it doesn't let you just consume data, but you can add to it as well. So if you're a chlamydia researcher and you're reading a paper about a new protein in a strain of chlamydia, you can, through these structured forms, you can actually add a new annotation for this protein. So there you have these forms that guide you through selecting a molecular function from the gene ontology and the termination method. You give it a reference, and then it creates a new statement in Wikidata. So once you do this, the statement is then immediately viewable in Wikidata and accessible through any other application that uses Wikidata as well. Lastly, Wikidata is incomplete. It obviously doesn't have every biological knowledge, biological database or ontology in it. But what I will talk about next is that it itself doesn't really need to be complete as long as it can be interoperable with other resources. So one way you can do that is through federation on Sparkle, through Sparkle. So federation is the ability to make one query across multiple sources. So if you have multiple Sparkle endpoints, that have URIs or unique IDs that they share in common, you can actually execute a query across these multiple Sparkle endpoints within one query. So this is an example of something you can run right now on Wikidata. So you can get drugs, information about drugs from Wikidata, and you can query for the pathways that these drugs are part of from Wiki Pathways, simply within the same query by executing certain part of that Sparkle query on this external service. So this is the Wiki Pathways Sparkle. Okay, so that's the end of the first part.